We're Desktop Genetics. We're a company based right here in London. We started up out in Cambridge in 2012. We actually used to work right on this floor about 3 years ago, so it's kind of cool coming back. We're a multidisciplinary company of biotechnologists, so lots of molecular biology, synthetic biology, but also software development, and we've applied those expertise to CRISPR genome editing, which I'm going to tell you a little bit about this evening. We work with some really, really cool companies and universities like Perello College. We have a grant with them to turn off the mechanisms of drug resistance in ovarian cancer. We're working with Illumina, which is the largest provider of sequencing technology in the world, to actually be able to read a patient's genome and then determine how we're going to modify it so that they won't have certain diseases anymore. We have two main paths to desktop genetics, right? Two products, or rather one product, one service. The product is the DeskGen genome editing platform. So if you've heard of CRISPR, I'm sure many of you have. It's one of these buzzwords that's floating around in the tech/biotech scene at the moment. Basically, it's a tool that allows you to go in and precisely modify any gene you want. You can add it, you can remove it, you can modify it in these very interesting ways. And there's literally dozens, if not hundreds of ways of doing this. Lots of different tools out there. We put it all in one place in the DeskGen platform, so we unite it. And then we can also design experiments for people and get them executed as well. So the product effectively allows you to develop your own CRISPR experiment, whereas our service, we design these experiments for people. That's what we have today. Where we're going is we're building an AI that can perform surgery on the human genome. So basically, if you have a disease, then we could literally perform surgery on the gene causing that disease so that you could actually get rid of something like retinoblastoma, or you could perform cell therapies where you go in and modify a cell to do something different. And we're very, very, very excited about this. So I want to give you a very brief introduction to CRISPR and how it works. I hope you all leave being able to explain the molecular biology of it. I want to tell you a little bit about how we sort of started the company, which was completely different to what we're doing today, and we had this massive, I guess, history of disruption over our 4 years, and how we plan on taking CRISPR into the clinic. So let's jump right in. Editing the genome with CRISPR. So MIT Tech Review, they like to publish this, right? Wired published these big headlines where they say, we can engineer the human race, the end of life as we know it. These are actual headlines on Wired and Tech Review. And that is true, it's happening, but it's not happening tomorrow, right? We're not all going to have superpowers. Rather, it's more like the applications that we see today are these gene or cell therapies, which you see here on the right, where effectively you can deliver the fix for a faulty gene using a modified virus directly into a human patient, or a cell or an animal model so that you can do research. Or alternatively, you extract a cell, like one of your immune cells, and we modify it to go after cancer or after a particular infectious disease. And this is how it's possible. You have an enzyme, right? So it's a big protein called Cas9, which likes to chop up DNA. It's called a nuclease. The problem is Cas9 itself is kind of stupid. It won't go anywhere unless you specifically tell it where to go. And you do that using this programmable RNA, which is just an acid like DNA. And what happens is these two things, the RNA is programmable in that you synthesize it chemically. And it has this variable region which will attach to any piece of DNA that you have in your cells. And it's also got this constant region which allows the programmable region to bind to the nuclease. So you get this complex of basically scissors that can cut DNA and a small RNA component that tells it where to go. And you can be very, very specific about where it goes in the genome. You can make a cut, and then you can delete a gene or you can insert a gene. And so this is what's actually going on in the cell, according to some authors. So that's the Cas9 docking, and the gene, your DNA unwinds, and then the RNA, that red bit, is binding to your DNA. It's checking to see if there's a match, and the Cas9, once it sees that there is a match, will then cut the gene. And then at this point, the cell is like, I don't like this, this is not cool, and it will try to repair the gene. And because there's no repair template, there's nothing there to tell it how to do it, it repairs it inappropriately, it goes wrong. And so effectively, this is like introducing an error into your code if you write software. The other way you do this is at the same time as you insert your CRISPR components, you insert a new piece of DNA called the repair template, And because it's sufficiently matched, oh, the GIF wasn't there. I had a wonderful thing where you could see the new piece of DNA come in and attach. But this is called a knock-in, where you actually knock in a new piece of DNA. And I just want to take a step back and say that this tool, this research tool, this medical tool came from basic research. Right? This was actually a bacterial immune system, where bacteria sort of do this internally to target viruses that attack them. And the only reason we have this tool, which could literally change life on this entire planet, is because some people wanted to study how bacteria defend themselves against viruses. So if anyone's trying to get rid of basic funding, tell them no, because this is a great example of why. So why edit genomes practically? Well, there's some cool stuff you can do with animals. Recently, a group looked at humanizing pig tissue by removing all of the viruses in a pig that could possibly target human, which would make their organs better suited for transplantation. Alternatively, if you're into farming animals, then you can make your animals resistant to pests and disease. You can make custom mouse models where you basically model a patient's cancer in a cell, in a mouse. You look at what the cancer is that they have inside of them, you look at the genes that make that cancer happen, you put it back into a mouse, effectively you recapitulate the disease, and then you can say, well, now this mouse has this disease, I know for sure that these genes, these mutations caused it, and now I can test drugs on this mouse and see what will fix it. Already, CRISPR itself has been used to literally delete HIV from human cells. It's been used for some fantastic immuno-oncology trials where these immune cells have been modified to go after a patient's cancer, and other gene editing technologies, which aren't CRISPR but which are sort of its forebears, have already had some very promising results. Just last year, a child here in the UK at Great Ormond Street Hospital was cured of cancer, completely gone, and she's still in remission, which is wonderful. And it's also been used to cure a patient previously of HIV. So this is some really, really cool stuff. Today, mostly, it's a research tool, right? People are using it in the lab to take a cell, turn off a gene, and then see what happens to that cell so that we can effectively understand what that gene does. So it's about really, right now, understanding how we work. But why start a business? Well, there's 3 million research experiments happening globally, which we could help with our technology. More importantly, there's 75 million patients which we could bring this technology to in the next 15 years. So how did we get to where we are in products and services and things like this? It's been quite the journey. Look at these fresh-faced people at the University of Cambridge. I know we all laugh when we see this. We met at the University of Cambridge on a course which was designed for people to disrupt the molecular biology and the pharmaceutical industry. And my co-founders, Riley and Victor, all agreed that we didn't want to start a drugs company, because if you want to start a drugs company, you're basically putting thousands of barriers in front of you from day one. Because a pharmaceutical company is basically a bet on whether or not a chemical is going to cure a disease. And you have to raise literally billions and billions of dollars to determine if your chemical works, if it doesn't kill people, if it doesn't kill animals in your early testing. And then once you get into these phase 3 clinical trials, you're literally spending millions of dollars every day testing a chemical in thousands of patients and hoping that it works. And that's very expensive. And we agreed, well, you know what, let's focus on the tools for the industry. And we came up with the idea of the synthbot, which was what investors later termed the biotech espresso. The idea was that when you're dealing with DNA in a lab, you've got some DNA that you've worked on in the past, and you've got some DNA that you've worked on in the past. And now I want to make something that is a composite of this DNA. Now, I have no idea that this exists in my organization, that I could take some of yours and some of yours and mix it together. So we built a search engine tool which could search for pre-existing bits of DNA, research molecules in the lab, and then it would say, well, you need something from this person, something from this person, put it into this machine, and it will remix the DNA into what's called a vector. And that's basically a functional element of DNA that you can use for research. And the whole idea was we would sell hardware, subscriptions to the software, and then the cartridges that would contain all of the reagents. Investors hated it. They said, hardware is like selling Ferraris, you're absolutely crazy. It's not a good thing because you have to go and design it, you have to build it, you have to ship it, you have to test drive it, you have to service it, but we like your software idea, that's scalable, so keep going with that. And so we did, and that was the auto-clone system. Cloning is a term which you might have heard from Dolly the sheep, but in reality, it's more commonly used for cloning bits of DNA, right? This idea of making new bits of DNA from what you have already. And we did that, and that worked, and then we got a grant, and the grant was exactly like how you said. It was a lot of admin and management and meetings, and it wasn't particularly fun, but it did help us prove our technology worked for finding pieces of DNA in an organization. And that's when we went out to a company called Horizon, and they said, hey, yeah, we build a lot of these DNA vectors, but there's this thing called CRISPR that has just come out, and there's this tool on the MIT website which we use every day, and it takes us literally hours to design an experiment, sometimes even up to a week. And the problem we have is we need to find these guides, these valid regions that can bind to a Cas9 so that we can cut the genome and do cool stuff with it. We said, well, we've got some technology that we could repurpose for this, and they commissioned us to build a tool called the Horizon CRISPR tool. And that was our big pivot from initially hardware to cloning software, and now suddenly CRISPR. And we retained the intellectual property on that tool, and we released it as a co-marketed tool with them called Guidebook. And that was half successful. We learned a lot about building custom tools for scientists. The most important thing when you're working with a scientist is you have to give them an abundance of data. Right? You have to tell them exactly why you're calculating something in the way that you're calculating it. And you have to give them all of the sources and all of the information. And if you release an update, you definitely have to keep the old software available, because they've just put months and months of work on that. And so, yeah, be careful with that. In any case, we kept going with this. We provided access to Horizon's customers to some of their reagents that they could sell through the platform. And then we brought together AutoClone, that old system, and the CRISPR experiments platform as DeskGen, which we launched in 2015. And we effectively united this tool set. We were very happy with it. And at this point, we realized, wow, we know a lot about CRISPR. And we know about everything you need to design and execute a CRISPR experiment, and we have relationships with a lot of partners. Why don't we start designing experiments and running them for people, and doing these CRISPR libraries as a service? And that's what we called Class. And so we started working with even more partners and folding them in, and then we released the second version of DeskGen. It's your idea of designing for the user experience in mind from day one, which we hadn't done. We had to go back and redo it again. But when we did, it was very powerful, because the convention for this kind of work is you have a form on the internet, and it feels a lot like you're stuck in 2002 for most CRISPR software tools. So we've had a tremendous reception with this very rich user interface. And now as we accelerate, we're bringing in more and more companies so that we can effectively execute what we call literal desktop genetics, where we go from designing the guides based around the DNA sequence of the cell that the user is working on. Because right now, most people work on what's called a reference genome, which is like all of our genomes averaged out, which doesn't really exist, no one has it. But then if you try and design guides using the reference genome, and really you're working on a cancer cell, which is like horribly, horribly messed up. It's got extra copies of genes, whole tracts of chromosomes are missing, and this can like really severely impact your in silico prediction. So we work with Illumina now to sequence the cell. Then we can design either this library of guides, many, many guides, or just a few. We work with Twist, who synthesize the DNA. Transcriptic, who are this great company out in California, who have a cloud lab. It's basically a big robot the size of this room with liquid handling robotics and molecular biology machines, and it automates this whole process of molecular cloning, which we have built AutoClone to deal with. We have a partnership with Vectalis, who can actually put our CRISPR molecules into a virus to more effectively target a cell, so that then the virus infects the cell, and it hacks its own DNA and can delete its own genes or add in new genes. And then we have this partnership with Horizon, where they can do the experiment for us. And we analyze that data and then get it back to the customer. So we're really, really excited about this, that we can effectively design an experiment and execute it without the biologist stepping into the lab. And we think about this a little bit as bringing the same sort of benefits the cloud has had in software to the lab. Because let's face it, if you guys wanted to start a software company today, you probably could, right? You hire some people in other countries if you needed to. If you had to build something cheap, you don't have to invest in all this capital equipment. You don't need server farms. You put it all on Amazon Web Services. And in biotech, you don't really have that yet, but it's coming, and this is very exciting. So we also work with Editas Medicine, which is a company that's actually running clinical studies of CRISPR in patients today, and the aforementioned grant with Perello, where we're using a modified CRISPR to turn off mechanisms of chemotherapy resistance in these ovarian cancers. So I had some notes which I just want to pull up, which I made for your talks. So yeah, Shelley, you said that the academic science culture is not permissive for this kind of creativity, right? Not as permissive, but in the startup environment it truly is, because we had no idea what we were doing when we go back and look at ourselves in 2012. We thought we had a good idea, but we changed it as I've laid it out a couple times, but in reality it was probably hundreds of times, right? We went through so many different cycles of thinking and speaking with people and finding out from our customers, or people we wanted to be our customers, what we needed to change. And I do also want to emphasize this idea of design rigor, right? One of the reasons that the platform is so successful is because we spend time doing user experience testing and just testing our assumptions endlessly. So yeah, good points, guys. Where's CRISPR going? So it's an international thing. There's a lot of countries doing cool stuff. So China this year, last month, just announced that it's doing a clinical study in humans for non-small cell carcinoma. States is doing a clinical study into CRISPR for sarcoma and melanoma. And in the UK, we have a scientist who is modifying non-viable human embryos to determine rates at how to increase implantation of an embryo in IVF. So she's not actually editing a human, but she is testing non-viable human embryos. And she has special permission from the government to do this to try and enhance pregnancy rates in infertile women. So very, very cool. We've released a new tool called Guide Picker, which is this awesome visual system for pulling together about 10 different scoring functions, each from a different academic tool, so that our clients and our academic users, it's free for academics, can basically use the state-of-the-art to design their experiments. And we keep this updated all the time. And then the next big thing is integrating even more with next-generation sequencing pipelines. And what that looks like is we sequence a patient's DNA, we look at real-world medical outcomes, and we look at biological data sets from public data, right? So this would be things like the 100,000 Genomes Library project, or the 1 million Genomes project out in the US. And all of this data is getting fed into our AI so that we can build, effectively, custom gene therapies for patients one day. But today, the focus is really in the lab. What we've demonstrated so far with the limited data that we've got over the last few years, and we're really scaling up to get more, is that our AI is about 3 times better than manual design. And this was from data a year ago. We're exploring more data sets, and we're very, very proud.